All right, a big thanks to everyone for coming for this month's PNRI Science Matters. We're very excited to have Dr. Scott Younger here joining us. Scott is currently the Director of Disease Gene Engineering at Children's Mercy Hospital in Kansas City. Scott did his training in a very illustrious cohort of graduate students with me at the University of Texas Southwestern Medical Center in Dallas, where he worked with David Corey on antisense oligonucleotides. He then did a postdoc with John Wren at Harvard, and then he worked in a really interesting group of scientists at the Broad in the genetic perturbations group. Scott's group now has what I would say is a really unique and amazing transition from thinking about newly discovered variants in a clinical cohort of pediatric patients in Kansas City, moving all the way through these amazing translational technologies that he's built in his lab to look at organoids and other cells from these patients for really understanding functional changes in phenotypes that are being caused by these genetic variants. So thank you very much for coming, Scott. We're excited to hear about your work.
Great. Thanks, Rick, for the very kind introduction. It's been great to catch up. And for those of you that I've talked to and will, I look forward to that. Here, let me share my screen real quick. Let's do that one. Okay, are we seeing it?
Yeah, if you want to project. Yeah.
That's it? There we go. Got it?
Looks good now. Yes.
Okay,
thanks.
Great. Yeah, so today I'll be talking about what we've really been focused on for the last 18 months or so, which is scalable approaches for modeling rare disease in organoid systems. So today's talk will kind of dabble in a few different things. We'll talk about discovery biology and linking that to translational research. We'll spend some time talking about personalized therapeutics and then on to precision therapeutics. So to start with genes and phenotypes. So the human genome is composed of 46 chromosomes. We have 2 copies of 22 autosomes and 2 sex chromosomes. And then distributed across these chromosomes, roughly 20,000 protein coding genes, right? And the spatiotemporal expression of these genes give rise to phenotypes. We think about these in the lab a lot as cellular phenotypes like proliferation, but cells make up organs and organs make up bodies. So really variations in the genome influence human physiology and disease. Here's an example of this from my lab, and this is really a poignant demonstration. Here, what we're looking at in these wells are embryoid bodies. If you're not familiar with this, this is what you get when you take a lot of some induced pluripotent stem cells and you put them together in a well. You'll form these 3-dimensional pluripotent balls of cells, and these are embryoid bodies. On the top, you're looking at an embryoid body that's been generated from a healthy individual, or what we call unaffected individual. And on the bottom is one of the patients here in our hospital. It has a rare disease. It's been diagnosed, and we know it's due to a single base change. And when you make an embryoid body from this patient, you can see right from the start that they don't make these healthy embryoid bodies. And we see this a lot with our rare disease patients. So you can see really at this cellular level that things are not working appropriately. At our hospital a few years ago, we launched this initiative. It's one of the flagship initiatives of our research institute. It's a program called Genomic Answers for Kids. And in this program, we're aiming to sequence the genomes of roughly 30,000 children and the parents, with children impacted by genetic conditions. This is largely, but not exclusively, undiagnosed rare disease. And the idea here is that we're going to build this large, publicly available data repository and also work towards achieving a diagnosis for these patients. So that's kind of where my lab comes in, is the research arm of this Genomic Answers for Kids program. So this is a really powerful warehouse or storage of data. But one of the challenges that we face with using this information to diagnose patients is just the massive amount of genetic variation that we have in humans. So between 2 non-consanguous individuals, if we're just talking about single nucleotide variants, we have on the order of 4 to 5 million differences between 2 individuals. And of those, on the order of a couple 100,000 are what we would consider rare. We focus a lot on rare variants because we anticipate that if a variant's causing a very rare disease, it's probably not going to be observed in much of the population. And now if we focus on offspring of these two individuals or a child, you'll detect on the order of hundreds of de novo variants, basically variants that are unique to the child. And we also focus on these quite a bit because we have two healthy, unaffected parents that have a child that has a rare disease. Many of these diseases we can think are monogenic. There's perhaps a higher likelihood that it's caused by a rare de novo variant in the patient. Okay, so we have this huge repository and we're aiming to sequence 30,000 children. And even if we just focus on de novo variants in a child, we're still talking about hundreds of variants across tens of thousands of patients. So how can we efficiently profile the functional consequences of all of these variants in a way that informs us about a patient's disease? So that's really the challenge that my lab is focused on tackling, kind of a population level of rare disease. Okay. So to get at this, it's helpful for me to think about this in kind of this gear schematic as the underlying genetics of disease, where we have a genetic sequence, and that informs a molecular process. You can think about this as gene expression or RNA splicing, transcription, and the ultimate result of all this gene expression is some type of cellular phenotype. So genetic sequences, that's what we have here in our Genomic Answers for Kids program. We're generating all this data, but there's plenty of publicly available data sets out there where you can go and you can collect this information if you're interested in studying. And really one of the big things that we're interested in now, at least for this first part of the talk, is understanding how those genetic sequences or changes in the genome influence cellular phenotypes. And the way that we're doing that in my lab is through high throughput pooled CRISPR screening. So studying gene function and associating that function back to genetic variants we're detecting in our patient population. Okay, so one of the really powerful things we can do with our cohort is we can take our clinical presentations of all the patients that are enrolled and we can stratify those out and bin them and see what's the most prevalent types of clinical presentations that we see. And this is from an earlier snapshot. I think we're up to just about 10,000 patients now. But the majority of these patients have some type of neurodevelopmental phenotype, but you can see here, this type of data really informs us as to what's most prevalent and what is likely, what areas we're likely to find genetic variants that are causing the disease, and also the largest portion of the patient population will actually be able to impact. So for today's talk, we're going to focus quite a bit on the cardiovascular system, or specifically cardiac development, and I think you'll see why clearly here in the next few slides. But now what we can do is we can take this whole patient cohort and we can say, okay, 582, and granted the number's larger now, but just to understand the approach, we can take this patient population and then we can just say what's going on in these patients, and we can focus specifically, and we've chosen to here, on morphological cardiac or congenital heart defects. And so for 184 probands that have morphological cardiac abnormalities, we can break that down even further into specific types of morphology defects. Okay, and so now we have those listed out. We can go into the individual genome sequences. And again, for this whole talk today, we'll only be focusing on single nucleotide variation. But in this set of probands, we have 184 patients. We identify 4,089 loss of function variants across 2,300 genes. So you could go one by one trying to study each of these genes and determining what their role is in cardiac development, but that would take many lifetimes over. So the strategy we're using is this pooled CRISPR screening. And the flavor of CRISPR we're using is CRISPR interference. And so if you're not familiar, this is a catalytically inactive Cas9 enzyme fused to a Crab repressive domain. So you can take a guide RNA and park this complex down on a gene promoter and repress transcription and basically inhibit gene expression. Okay, so if you, in these screens, and I should say we're also using CRISPR-I, it's been shown to be a little bit more effective and robust in stem cells than knockout, and that's why we're choosing that modality. So in CRISPR screening, what we're doing is generating a large library of guides. Our library is 60,000 guides, 3 guides per gene. And then we can transduce a cell population in a manner where each cell gets 1 guide. And then we apply some selective pressure. Typically, this is done in like a 2D cell culture, where maybe you'll apply some drug and look for drug resistance or look for genes that impact proliferation. But we're really interested in studying development. And so what we decided to do was kind of a wild idea of doing pooled CRISPR screens in organoid models. There are probably more reasons to think of why this experiment would not work than to why it would. Most of the screens that have been done to date are done in, like I mentioned, 2D cultures of homogeneous cell population that you're looking for cell autonomous gene knockout effects. Whereas here we're talking about a 3-dimensional ball of different cell types interacting with one another. So you lose that cell autonomous feature, and it was unclear if this approach was actually going to work. Now, I should say also, if you're not familiar with organoids, these are essentially 3D cellular models that mimic many features of development. So we figured perhaps we'd be able to screen in these organoids and find developmental regulators. So here's just an image of cardiac organoids at 2 weeks, and this is really one of the driving reasons for why we started out in the cardiac space, because you can really convince yourself you've made the right thing because it moves in a dish. So you can actually see these things contracting. It's bright field on the left. And then you can incubate them with a calcium indicator dye on the right here, and you can see this calcium flux over the organoid, and that's coupled to contraction. So you can really do some cool functional stuff with these organoids. Okay. So we took a huge population of iPSCs, we transduced the library in, and then we took them through cardiac organoid generation. And here I'm showing you kind of the summary statistics from that screen. So this graph is showing the guide RNA abundance in our CRISPR library. So, and this is normalized read count. So that first plot here is showing the distribution of guides and the actual pDNA that's used to make the library virus. And then we're looking at the distribution of guides at day 7 and day 14 when these are actually mature organoids. And what you're seeing is that distribution widening out. And that's really promising. That's happening because certain genes or certain guides are enriching in the population, others are depleting, which means there's some functional impact of knocking down the genes that we're targeting. Here is replicate reproducibility of these screens, and this is a big one. Again, we typically see really great concordance in 2D cultures, but we've never done this in organoids. But what we're seeing is we can recapitulate the effects of knockdown across replicates. Now, this is a busy plot, but the dots that are labeled in red are guides that target genes that we refer to as common essentials. These are genes that, based on hundreds and hundreds of screens, we know are required for every cell type to survive. And then in blue is the non-essentials. These are genes that essentially that you can knock out, and they don't seem to really have an impact on any of the cell models that have been screened to date. And then the rest of the points are just the other guides in the library. Great. Okay, so here's a busy plot, but it's pretty a systematic breakdown of some data. So here, each box represents a gene, and each box plot in there represents the depletion of guides targeting that gene. So I mentioned we did this in cardiac organoids. We also did it in iPSCs, just cultured for 2 weeks, and also in those embryoid bodies, that intermediate state. And we did this because we wanted to make sure we were identifying genes that were required for cardiac organoid formation, and not just in general for cells to survive. And so you can see in many of these boxes, genes that are, or guides targeting genes that are specifically depleted out of the cardiac organoid screen. And if you're looking in, you see that these genes, they all start with this NDUF, and that's because they're all in the same gene family. These are mitochondrial complex 1 subunits. And this is promising because these genes have known associations with cardiomyopathies. I wouldn't say it's a slam dunk, but it tells us that the screen seems to be recapitulating at least a little bit of biology that makes sense. So now that we have kind of genome scale data, we can go back and look at the gene set enrichment analysis. And so we can do this for different gene sets. Here's an analysis of developmental regulators. So again, a busy plot, but the red points are from the cardiac screen, blue are from the EB screen, green is from the iPSC screen. And so we're looking at normalized enrichment scores from gene set enrichment. And so what really sticks out is in our cardiac organoid screen, the gene sets that are enriching out are circulatory system, tube development, heart development, vasculature, all things that really make sense with cardiac organoids. So this added some extra confidence that these screens are actually able to identify important genes. I should say, with a big grain of salt, like there are some master regulators of cardiac development that didn't come out in this screen. So I wouldn't say these are comprehensive in terms of what they identify, but they can be really informative. And then just to go a step further, we can do this type of gene set enrichment analysis, but we can look at disease relevant gene sets. And so we'll wade into this slowly. Here we're looking at, actually, this is gene expression data from GTEx, just to kind of see what comes out of here. And so from the GTEx data from the ventricle, you see enrichment of ventricle morphology gene sets, and then cardiomyopathies and hypertrophies, which are kind of like heart function. So that makes sense that these things would come out of that data set. Surprisingly, in the GTEx atrial set, you didn't see much enrichment of anything. Here's the enrichment of genes from the CRISPR screen. So the first two columns are from expression data. These next two columns are from CRISPR screens and iPSCs and embryoid bodies. Again, you really don't see many disease gene sets popping out. But when we look at the cardiac organoid screen, all of these cardiac morphology disease gene sets pop out. And this was really, really striking because this is exactly why we wanted to do this screen in hopes of identifying genes that impact heart morphology. So we're really, really confident that we're going to be able to identify some novel genes in this data set. Just a snapshot. I'm not going to show a lot about this, but we're in the process right now of validating these CRISPR screens. So here is, again, each box plot is an individual gene, and you're looking at depletion of guides targeting that gene across the three screens I've been talking about. You'll see that the genes deplete out a little bit more in iPSCs, and that's because those cells are proliferating, whereas the EBs and the cardiac organoids are kind of more of a differentiating, and they have less proliferative, less proliferation going on and less opportunity to deplete. And again, those two genes are common essentials that we know are depleted out of all screens. Here's an example of a gene that was depleted specifically out of the cardiac screen, and then we see a number of these, which are just really interesting genes that kind of jackpot in the organoid screen and look to enhance differentiation. So that's one we were trying to understand what that could mean. So we've gone on to the individual validations. Again, here's a cardiac organoid. So cardiac troponin is a marker we use to ensure that this is an actual cardiac organoid. But if we knock out that gene, we can never get these guys to go through all the way to cardiac differentiation. They actually form these balls and they just keep growing and growing and growing. And then here's an example of a flow-4 stain where you can see in a control organoid, you see that calcium flux and that movement, and those, if you knock down this gene, they just kind of sit there. So we're in the process of going through kind of a few of these high-profile hits and understanding again how they may be impacting cardiac, at least organoid formation. But I want to, as we're doing that, I'll take a step back and look bigger picture of, why are we doing this? So as I mentioned, we have all of this genome sequencing and these electronic medical records, so we can go look at our patients that have, cardiac or congenital heart defects or structural defects, and look at their genome sequencing and identify loss of function genes. Now on the right here, this plot, again, busy, but what we're looking at is guide depletion. So this is all guides in the library. You can see that they bulk up right around 0 in terms of log fold change in the screen. This second column is non-targeting controls. Again, they target 0. And then this plot, every row represents an individual gene, and those 3 tick marks represent how far out the guides depleted or enriched, the guides targeting that respective gene. So one thing pops out right away. One of the top hits is this zinc finger 69. This gene is virtually unstudied. I don't think there's any papers on it, but from our data, it's one of the most required genes for cardiac organoid formation. So we can go back and we can find patients, or at least this patient in our patient cohort, that has a loss of function variant. It's an undiagnosed patient, the loss of function variant in this gene, and you can see that they do have these cardiovascular phenotypes. Now, there's a lot more work to tie these in and say that this might be a causal gene, but these are the types of projects that are falling out of this approach, and it's not the only one. So this is, again, the discovery biology component of what we're talking about. And each of these will develop on a project that we can pursue further. So now we're going to take a little bit of a transition. In the original inception of these projects, one of the things we were interested in doing was generating cell models where we could try and model these changes. And the original idea was to take a commercial stem cell line, engineer in potentially pathogenic variants, and see if we could recapitulate phenotypes. But that turns out to be incredibly inefficient, and we can't really scale that approach. And what turns out to be way more efficient for us, at least, is generating patient-derived cellular models that have that genetic background baked in. And to do that, though, has been quite the task. So when our patients enroll in the program, they provide a standard blood draw. And this is what we use to isolate genomic DNA and perform genome sequencing. And when they enroll, we save some residual PBMCs, and we'll use these for various expression profiling experiments, but you can learn a lot by seeing a way a gene's expressed, either its level of expression or perhaps a transcript structure, and that can be informative and help achieve a diagnosis. But in general, these blood cells only express about 20% of the transcriptome, and if you're not looking at a blood disorder, the likelihood of finding anything interesting or informative is pretty low. So we've gone pretty much just to biobanking these things. So for all of our patients, we have these residual PBMCs stored in a cryo tank. And we realize this could be a great source for generating patient-derived iPSCs. But this is no small task. So just for context, you can do this with approaches like Sendai virus to reprogram, but that costs thousands of dollars per sample. So it's not scalable. There are some CROs that'll do this, and there's some places that'll do it for free, but the turnaround time is typically a year to 18 months till you actually get something back. And for the ones that you pay for, you'll pay up to like $10,000. And they require quite a bit of material to get this through the pipeline. So we're in a spot where we're doing this on an academic, funding structure. So we don't really have those kind of resources. And we don't have enough cells to do this. So through a Herculean effort, a very talented research scientist in the lab generated this very straightforward protocol, at least on paper, where we take standard reprogramming factors. We actually found a nucleoeffector, a decommissioned nucleoeffector in an old lab. And we were able to get this protocol to work very robustly, where we can generate patient-derived cell lines in about 2 to 3 weeks for about $200. Excuse me. So this makes it possible for us to start generating lots of patient-derived models. The other thing, because we're working with very small numbers of cells, we're working in small volumes, and small volumes are naturally amenable to scaling. So we do our reprogrammings in one well of a 12-well dish, but we can take 12 samples and do them in parallel. So in one dish, we can generate up to 12 patient-derived cell lines. It takes about 2 to 3 weeks. We biobank those, and for some of those, we move on to characterization. So this slide is somewhat dated. We've generated well north of 300 patient-derived cell models. And what I'm showing you here is our success and failure rates. So at the time of this slide, if we had over 300,000 cells, we had a 100% success rate with reprogramming. This is important. One of the things we were worried about is there would be some intrinsic feature of a disease that would preclude its ability to be reprogrammed, but we're simply not seeing that. And through further optimization, we're to the point now where we've been able to robustly generate iPSC lines as long as we have at least 5,000 PBMCs. Great. So, when we started this, we wanted to generate these models, and we wanted to kind of study diseases, and we wanted to use a technology where we could perturb gene expression to kind of study disease. For a lot of reasons, CRISPR is challenging to work with in stem cell models, so we resorted back to these antisense oligonucleotides. As Rick mentioned at the introduction, I had some experience with these from graduate school, and they've had somewhat of a renaissance in the clinic, so we thought it would be a good approach. So here I'm going to show you just some data of how we got up and running with these ASOs. So again, our workflow here is we're taking these induced pluripotent stem cells, and you form these into these three-dimensional embryoid bodies, and then you differentiate them further into cardiac organoids. So we picked a target gene. This is cardiac troponin. So this is a marker for cardiac cells. So we knew it was expressed, and it's also required for contraction. So we knew that if we could effectively knock this gene down, that we would see an observable phenotype. So again, if you're not familiar with antisense oligonucleotides, they're synthetic nucleic acids, about 20 to 30 nucleotides in length. They recognize complementary RNA sequences, and there's a few different modalities through which you can interfere gene expression. The ones we're using are just steric blocks, so you can target the translation start site and prevent translation, or these splice donor acceptor sites and prevent splicing. So here again, this is a cardiac organoid treated with a negative control ASO. You can see that robust troponin staining. But if we target any of those three sites that I mentioned, we see what looks to be a complete ablation of troponin expression. So really robust knockdown, which we were happy with. The other thing we looked at was contraction. So we grew these organoids in these agar well dishes. So these are dishes, and in each well they have many grooves. So you actually generate hundreds and hundreds of organoids in each well. So it makes it really easy to say, okay, let's profile 100 organoids and see how many are beating. And if we knock down this cardiac troponin, we can see we completely knock out or inhibit contraction. Okay, so somewhat of a left turn, but while we were doing these experiments, we were doing some ASO stuff because we were interested in using them. We were generating these patient-derived cell lines. And then we realized that in the process of generating these lines, we had done a small cohort of Duchenne muscular dystrophy patients. So these fall in the bin of patients in our program that are diagnosed. This particular patient has a structural, so Duchenne muscular dystrophy is caused by mutations in the dystrophin gene. This patient has a structural deletion of exons 46 through 53. And so what you get is an exon 45 connecting to 54, and that results in a frame shift and no productive dystrophin expression. Okay, so this caught our attention because just a couple years ago, the FDA approved an ASO therapeutic for the treatment of Duchenne muscular dystrophy. This is referred to as an exon 45 skipping ASO. So the way it works is it prevents splicing to exon 45. So now 44 is going to connect to 54. You can't gain back that genetic information, but you do end up with an in-frame transcript, and that dystrophin that gets made is therapeutically beneficial. So we were curious, can we generate cardiac organoids from this patient? And what happens if we treat those with an ASO matching the sequence of that FDA-approved ASO? So I'll say for any of the ASOs I'm talking about today, these are fully 2'-O-methyl modified RNAs. They're not the exact drug that goes into people, but they match the sequence of the drug. Okay, so here is a cardiac organoid from an unaffected individual. So in green, you're seeing the dystrophin staining, and in red, you're seeing the troponin. So we know this is a cardiac organoid. And here's what happens if we generate a cardiac organoid from that patient. You can see the dystrophin is completely absent, but this is a cardiac organoid. But if we excuse me, if we treat those organoids, the mature organoids with the ASO, we see a robust restoration of dystrophin expression, quite striking. And as it turns out, there's a second ASO that's in, I think it's phase 2 clinical trials, another exon 45 skipping ASO. This is a popular target because about 8% of DMD patients fall under the umbrella of being amenable for this therapy. So this second drug we tested, or the second sequence, and you see again robust restoration of dystrophin expression. So here is a cardiac organoid from an unaffected individual. You can see it sits in a dish and it contracts. But if you generate a cardiac organoid from one of those patients, or from that patient, excuse me, you can see it just kind of sits there. It will eventually contract, and you can see some cell debris moving in there, so you know it's a video, but they don't really contract. But when we treat with that FDA-approved ASO, you see this restoration of contraction, which is quite striking. You can observe just through a light microscope the functional restoration of the dystrophin. And again, this is that second ASO is in phase 2 clinical trials, and we're seeing functional restoration of dystrophin. So this had us really, really excited that we were able to profile, a known drug and see a known response in a patient sample. Obviously, had we not known that, or if these were not FDA-approved drugs or in clinical trials, we would be very excited about pursuing these as possible therapeutic leads. And that had us turning back to our patient population and saying, are there any opportunities here? And as it turns out, we have another pair of DMD patients. These are siblings. DMD is an X-linked disorder. Both of these patients, little boys, inherited the X chromosome from their mom and the variant from their mom. And this variant is a deep intronic variant between exons 43 and 44. And this gives rise to a novel splice acceptor site, an incorporation of a cryptic exon and premature transcript termination. Again, these are diagnosed patients, so we know this is the causal variant. So we were curious, what would happen if we designed some ASOs to target that site? Could we restore proper splicing, and could these be preclinical therapeutic leads for these patients? So we designed 2 ASOs. I mean, really straightforward design here. All we're doing is straddling right over that mutation that gives rise to the splice site and the incorporation of a cryptic exon. So here we'll call this patient Y. This is one of those siblings. Again, you can see a complete absence of dystrophin in the cardiac organoids generated from this patient. But both of the ASOs that we designed were able to restore dystrophin expression in the patient-derived organoids. And this is even more unique than, rare, is we have siblings here that have the same disease. So we have 2 genetically similar but independent models, and we can see this is the other sibling, and we can see restoration of dystrophin expressions. This is great. So here we're looking at these cardiac organoids incubated with this flow-4 calcium indicator. Again, you see this flux of calcium across the organoid, and you can see in the video, you can actually see the organoid moving. So it's coupled with contraction. Now, if we generate organoids from one of those patients, you see this calcium flux, but it's sporadic. It doesn't go over the whole organoid, and you can observe that it's completely decoupled from any contraction. So these aren't functioning like healthy organoids. But when we treat with either of those ASOs, we see, again, the robust restoration of this functional contraction, and you can even see that calcium flux being restored. So really, really exciting results. We're not the first people to go down this path, for patient-customized oligonucleotide therapies. Tim Yu at Boston Children's published a beautiful paper in New England Journal of Medicine in 2019, where they did this for a patient with a different disease. That patient had a condition called Batten syndrome. And I won't get into the weeds about how that disease is caused, but basically they had one allele that had a loss of function mutation, and then the other allele, there was a retrotransposon insertion that gave rise to a novel splice acceptor site. So the same variant consequence or insertion consequences we're seeing in our patient. And their strategy was to tile that splice site with that novel acceptor site with ASOs and see if they could restore splicing. And they did this in patient fibroblasts. Now they did treat the patient with this, so that condition is associated with seizures, and what they saw is a significant drop in the frequency and duration of seizures. So there's proof of principle for this approach actually working. So there's a little bit more to that. They had to formulate the ASOs, and the way they did that was by matching the chemical modifications and features of drugs that were already in clinical trials to treat the brain. And then you administer that intrathecally, and then it gets to the brain. But it sets this proof of principle where if people have already gone down this path of optimizing therapeutics and finding ways to try and get them to work, you can kind of borrow the modalities and the modifications to get something into the clinic. So the timeline for that paper that I'm referring to, from clinical diagnosis to when they were able to first dose the patient, was it was about 18 months that it took from start to finish. But from the data that I've shown you here today, we're able to go from PBMCs to iPSCs to organoids and ASO leads is about 6 weeks of hands-on time. So this project took us far longer than 6 weeks, but now that we have all these modules in place, we could repeat this whole process in about 6 weeks. We haven't made it obviously to the clinic yet, but we're working very quickly. And so we've really optimized and expedited the process of pursuing these personalized therapeutics. So this is really exciting. We refer to these as personalized or N of 1 therapeutics. But one of the things my lab is really focused on is scalability, and N of 1 and scalability kind of are like oil and water. But we kind of took a step back and said, what did we just do here? We took a disease, Duchenne muscular dystrophy. We generated a cell model, these cardiac organoids, and then we targeted a pathogenic intronic variant to see if we could reverse it. And so we distilled that down to kind of its basic components, just a phenotype, an assay, and a variant. So we realized if we focus on these 3 components, maybe we could scale up this approach. So phenotypes. We happen to be sitting on a large repository of data, so we can actually look at all of our patients. Again, the slide's dated, but these numbers kind of hold as the repository's grown, and we can say, what are the most frequent clinical indications that we're seeing across our whole rare disease patient population? And among the most frequent is seizures. Global developmental delay is always at top, but that's such a catch-all that it's really hard to know how to interpret that, at least in terms of developing a functional assay. But seizures are, enticing for a target because they're prevalent, and the proof of principle is already there that these personalized therapies can work for that condition. Okay, so now we need an assay, and this is one where we had to do some developing. And to do this, we're going to work in brain organoids. So it's very similar in that we start with iPSCs and generate these these embryoid bodies, but then we take them down a different path. We take them down to these complex cerebral organoids, and here you're seeing staining for a neuronal marker. And so we make these things, and again, we stain them with this calcium indicator dye, so we can use a lot of the same tools. And this is a video. You kind of have to watch it for a second, but I've circled in red where you should be looking. And this is a 4-week-old organoid, and you'll see here or there, it'll blip. There'll be a little bit of a signaling. So you can see it's low, but it happens. But if you do the same assay for a patient impacted by seizures, these are actually siblings. You can see that there's just a heavy and qualitative difference in neuronal activity. You can see these things are just going wild like a light show. I was skeptical that this was a real phenotype, but we've gone through. Here's another example of one where we have a patient, and this one has an unaffected twin and an unaffected sibling. And you can see in the affected individual, you can see there's just far more activity than in unaffected individuals. We've gone through at least 20 to 30 different patients, and all of them have exhibited this increased neuronal activity. So now we're confident that we have an assay where we can actually measure kind of seizure phenotypes. But those are actually the low-hanging fruit, or the easiest things to get at. The real challenge is identifying variants that we want to target. How do we identify splice-disrupting variants using genome sequencing data from a large cohort of patients? And so to do this, we've engineered a massively parallel mini-gene splicing assay. And that starts with just this basic mini-gene splicing assay where we have a GFP that's separated by a human intron that we cloned in. And in the middle of the intron is a cloning site that allows us to drop in any intronic sequence of interest. And we're focusing on sequences of about 100 nucleotides long. So if we clone in an intronic sequence and it has no impact on splicing, the GFP will express properly. You can PCR over the exon-exon junction and you'll see a clean product. But if that intronic sequence of interest disrupts splicing, you'll get this hybrid PCR product that has a mix of exonic and intronic sequence. So it's pretty basic in design. So we decided to test this out using that DMD variant that I told you about in that patient. So we took 100 nucleotide windows and dropped it into this mini gene assay. So these are two independent vectors. They're identical with the exception of one nucleotide right in the middle of this intron. So we put these in hex cells and you express it. And you can see for the reference sequence, you get a clean product. But with the variant allele, you get this extra band, or we believed was disrupted splicing, and then we sequence, and you can see this GFP exon 1 connecting right to the intronic sequence we cloned in. And what was really interesting is this matches to the nucleotide where we see disrupting in the actual patient. So this is a high fidelity recapitulation of a splicing defect. So we were really pleased with this result. But now, our interest is scalability. We want to profile lots and lots of variants. So what we're going to do is turn back to our patient cohort. We're going to look at all those with seizures, and we're going to pick out rare deep intronic variants. I guess not necessarily deep. We'll look at all intronic variants. We've done some filtering in terms of the frequency of the variants in the population, and also the function of the gene for loss of function and tolerance. And also, we're focusing initially on dominant inheritance because we believe it'll be easier to identify monogenic disruptions in dominantly inherited diseases. So the snapshot of this was, roughly 10,000 variants we were able to pull out of our whole data repository. And now we want to use this assay that I just told you about. So I mentioned profiling, one sequence at a time. But what we've done is engineered a multiplexed barcoding strategy that allows us to read out splicing outcomes using a next-gen sequencing strategy. And so here I'm showing you one of our assays where we profiled 4,600 variants. Again, instead of cloning in one variant, we print up libraries of tens of thousands of oligos using array-based sequencing, and then clone that into a large complex library, which we can toss into cells. And here's a metric. I won't go into it, but basically we're evaluating changes in splicing between variant and reference alleles. And in green, I'm showing variants where the variant allele gives rise to, or influences splicing acceptors. And in blue is novel splice donors. And what you can see, most variants don't have any impact on splicing as we would kind of anticipate, or that's the null hypothesis. But you see these outliers were variants that have a dramatic impact by giving rise to novel splice acceptor or donor sites. Digging through the data a little more, some of these outliers we spiked into the library. They were known pathogenic variants from the literature. I'm showing you those here, and you can see where those variants are in red and where those the cryptic exon is in blue. And one of the things that we saw, it was really fascinating that this assay reads out at single nucleotide resolution where these splicing disruptions are observed in the patients. So again, this is really high fidelity, and it's allowed us to go a little bit further, and we identify variants like these ones that I'm showing below, where the variant doesn't occur right at a splice site, but it occurs in a splice enhancing sequence, whether that be in the cryptic exon or farther within the intron. So you don't see the splicing aberration where the variant happens, but it's somewhere nearby. So this has been a really powerful tool for identifying these kinds of variants. So now we're at the stage where we have this patient cohort, we have these intronic variants, we have strong evidence that they disrupt splicing, at least in a reporter assay, but we have to go back in and validate these in the patients. So again, here is a brain organoid from an unaffected individual. I've circled in big red here where you should see some activity. It'll eventually go off, but there it was. But here we've gone through and we picked a selection of 8 patients interestingly, patient 8, H, I guess that would be, had structural defects of the brain, and that's the only patients thus far we have not been able to generate brain organoids for. They just won't develop. But for the other patients, we have them. You can observe here this seizure-like phenotype across all of the patients, which is fascinating. And I've labeled here, the variants that we detected splicing aberrations in our assay. I wish I had more to say about these. We're in the process of doing bulk RNA sequencing to validate those splicing aberrations in those patient-derived cell models. So we should know soon, actually, I think in Friday, if we can validate these. And for some small set of these, those will be diagnostic findings. Okay, so this is kind of an overview of what I've just shown for our personalized therapeutics kind of platform, where we do this whole genome sequencing, and using that data, we prioritize variants, using the massively parallel reporter assays. There's a little jump here. I've shown you how we profile that in brain organoids. We do have a component and a collaboration at the Stowers Institute, which is a local research organization here, where for genes that we identify variants that disrupt splicing, but the gene has no known function, we model those rapidly in zebrafish. And there are seizure phenotypes in zebrafish that we can see. And then we ultimately use these ASOs to evaluate or confirm the molecular impacts of the variant. And then you can see kind of in light gray here, the idea of going back to the patient with a therapeutic. That's in light gray because we still haven't gotten to that point yet. We're still trying to find ways where we can get to that final stretch. Okay, so that's kind of getting us through the personalized therapeutics. And now in the last minute or two, I just wanted to touch on the newest program we have, which is focused on precision therapeutics. So again, you saw this a little earlier. This is making brain organoids and profiling seizure activity. This has just been really interesting to see. And we just have so many patients to look at. We actually demoed a really nice confocal microscope where we're able to take these the organoids and do the calcium signaling, and you can focus in on particular regions, and then you can profile neuronal activity across the organoid. And we were really excited about this. And then when we looked back at it, this little 3-panel picture here took about 4 hours to generate at a microscope. And we realized that's not really scalable for what we're interested in doing, but we thought about it and said, hey, we're really just looking at light here. So we adapted this to a 96-well format. And the way we do this is we plate iPSCs in an individual well. So this is a U-bottom, and we generate brain organoids. So 1 organoid per well of a 96-well plate. And then we use this calcium indicator dye, but instead of using a microscope, we just use a plate reader. So in this layout here, each row of the plate represents an individual, and each column is, a replicate organoid from that individual. Here's a snapshot of data off the plate reader. So this will match the videos that you saw earlier, where you can see an unaffected individual. You'll see over a time course of about 100 seconds, you'll see a few blips. But when you look at patients with seizures, you can see dramatically increased activity. So this is really, really promising. So we decided to take this a step further and do some drug screening. And so here the layout's a little different. We've done unaffected individuals and patients, and now we're profiling basically untreated organoids. TTX, this is a tetrodotoxin, is a neurotoxin that shuts down neuronal activity. And then we tested a few different drugs, and these were really just things we could get pretty cheap from Sigma, as reagent grade powders. But they're all different seizure medications. So one of the reasons this is really powerful also for seizures, this is one of these indications where there's on the order of 28 clinic FDA approved anti-epileptics in the United States. Any given patient only responds to a subset of these, and there's really no way to know which ones will work. So it's a trial and error approach. And while you're going through that, you deal with the side effects of these drugs while you're waiting to see if it's going to work. We recently had a patient advocacy dinner where there was a mother that spoke about a daughter who required 7 years of different trials with anti-epileptics, till they found this a regimen that worked for her. So here we thought this would be great if we could just screen rapidly through drugs, and we were really surprised, happy, I guess, to find that we saw these patient-specific responses to various therapeutics. So again, the top row is an unaffected individual, and you see this low level of neuronal activity. And in all these, you see the tetrodotoxin totally knocks out neuronal activity. So we know what we're looking at here is actual neuronal firing. So you can see increased activity in each of the patients, but patient 1 only responded to drug A and C, patient 2 only responded to B and C, and patient 3 responded to A and B. And if we go back to the mechanisms of action for these drugs, which are shown over on the left, there's really no correlation with why those drugs, the different drugs, worked. And this is a small subset, but what we are seeing is patient-specific responses. So now we're in the process of going back to our patient cohort and finding the patients where we have the most clinical data for what they've been treated with. And we're going to repeat this and profile those actual drugs in those patient-derived models to see if this really does have the predictive power that we hope it does. And if it does, we're looking at a screen that takes about 8 weeks to do, and potentially under $1,000, rather than going through 7 years or so of clinical visits. So taking time away from the impacting the health of the child, the quality of life, impacting the health system, and just savings all around. So we're really excited about this and moving this platform forward. That's right where I'm going to stop. Just want to acknowledge the people in my group that did all this work. John Means is a very talented scientist who generated our iPSC pipeline and all of our organoid work. Jasmine is doing the CRISPR activities and the screening, and Annabelle generated the splicing assay. Our director is Tomi Pastanen. He's a very important person. He's the lead study investigator for Genomic Answers for Kids and makes this all work. And also Genomic Answers for Kids, it's easy to kind of gloss over that we have a bunch of data, but this is a small but mighty team of coordinators, genetic counselors, and bioinformaticians that make all this data available to labs like mine. Our work is funded by internal funding mechanisms through local nonprofits, BioNexus, and through NICHD. And this is a picture of our mothership here. So with that, I'm happy to take any questions.
Thanks very much, Scott. That was awesome. Tons of amazing data there.
Thanks.
If you have a question, feel free to raise your hand or type it into the chat box. Scott, I'm wondering about the power of the organoid system. Are there, for example, the seizure phenotype you're seeing with the calcium fluxes, can you see those in single cells, or are some of those phenotypes only manifest when you look at it collectively in the organoids?
That's a good question. You do see, if you do 2D differentiations, you do see these. So one of the reasons that we really got into the organoid space, we're working with these rare diseases, and we know there's neuronal phenotypes, but when we sat down and said, what kinds of cells are we going to make? If you go the 2D route, you've got to make some decisions. And we didn't know what to look for. So we figured the organoids would just be a way where lots of stuff gets made, and then we don't have to make that decision. So that's what got us in this space. So everything we've done has been in the organoids, but when we've done 2D, like straight neuronal differentiations for the seizures, we have seen those phenotypes as well.
That's cool. Other questions for Scott?
Don't be afraid to ask questions.
Hey, Scott.
Hi.
Wonderful work. Thank you again. My question is about the different types of transcripts that you are going directly to the organ, which is, I'm assuming that's one of the good reasons, right? Did you manage to look the type of transcript that you are looking into, or no?
I guess I'm not quite understanding the question.
So each gene may have more than one transcript, right? Like multiple different types. And going to the organ, it's easy in this way as well, right? Because you probably are dealing with the right transcript. So for the tissue.
Yeah.
I'm just wondering if you guys had a chance to look into the different transcripts or no. Like you have a mutation.
Yeah. When we chatted earlier, we kind of mentioned this. So we're looking at rare diseases, and these patients, as I showed you, saw one of their kind of like clinical record. Nearly every organ system of the body is impacted for many of these patients, and so these are really penetrant variants. And so we're really just focused on that exact site where we're seeing the variants and seeing if we can recapitulate that, and then in the organoid models reverse the phenotype. And we're not really looking outside of that in terms of, we don't even really know like what's the exact cell population where this is happening when we have the complex organoid. But if we can see a reversal of the phenotype, we know at least it's responsible for the thing that we were seeing. So it's kind of our first pass. And really, we're using this as kind of like the canary, right? So we want to see if we can see something happen functionally if we perturb that variant or reverse that variant. But we don't really go deeper. All those things are interesting and can be pursued after we know that the variant or the transcript does something.
Okay, thank you.
Scott, there's a couple questions in the chat. Are you able to see those?
Yeah, I see them. So stability of ASOs. So that's the question we're asking right now. We're doing this in our DMD model, and so that's great. And we've done this and had these things go out for that. It was 4 weeks that the effect lasted in a cell culture model before it reversed. But as it turns out, dystrophin is a really stable protein, so we don't know exactly how long the ASO was around. So we're doing that experiment now to see how long it lasts. I'm not reading too far into what that translates to clinically, but this is well worked out for the administration of these ASOs, the modifications to get them to last longer. So that can all be addressed if you're actually interested in pursuing therapeutics down the road. And also ASOs instead of CRISPR-I. So CRISPR, it turned out to be a pain. We did the screens. The screens were really challenging. But the CRISPR line or the iPSC line that we use to do the screens has the Cas9 engineered into a stable, one of these safe harbor loci. So it stays on. Now, if you just try and do a typical approach, like you do with CRISPR, like a lot of us do, where you just lentiviral put Cas9 in, iPSC shut it off. So that's why we've kind of abandoned CRISPR for that approach, because generating a Cas9 line, for every patient or something like that would just be too cumbersome and not scalable. So that's the reason we went ASOs. And then off-target effects with the ASOs. Again, we haven't seen that, but we haven't looked for it. But for the question we're asking, the purpose of the experiments, the purpose of our initiative has always been to generate a data repository, to see if we can achieve diagnoses for these patients. We never started the initiative with the intent of generating therapeutics. It wasn't there. The ASOs have they're just a modality where we use them to validate the splicing effects, the pathogenicity of a variant, but that molecule is just inherently a therapeutic lead. So we're not looking for off-target effects, because really we're just trying to see if that variant, if we can target it and reverse the effects, the functional effects of the variant in a diagnostic way. So we're only looking for the kind of the true positives. We're not really looking for what else is going on. If we were ever wanting to go further into the clinic, we'd have to move further into controlling for that.
So thinking a little more about maybe that clinical route, you mentioned it's really, really difficult to have some new sequence ASO. How many clinically or FDA approved ASOs are there out there that you might sort of farm for these?
Yeah. So great question. I think there's on the order, and my PhD advisor would be so mad at me for not knowing the exact number of ASO therapeutics that are in the clinic, but we're talking, dozens to dozens, like not a ton. But this is also another tough nut to crack. There's thought that, you take this RNA sequence is essentially what we're using, and we modify it, and different modifications at different nucleobases and different backbone modifications stabilize this thing and impact the PK/PD of the molecule. But it's thought that like there's a messy combinatorics problem there, where every change impacts every nucleobase differently based on its nearest neighbors. And so it's not going to be a perfect, like, just borrow that backbone and just swap the sequences. And that, I mean, people are working on that, and it's a very, active area, but I'm letting them figure that out because I'm just not the person that's going to be able to solve that problem. But yeah, it does get really challenging to find. There's no magic bullet that's just going to be a perfect solution for all of these ASOs.
Amy, you have a question?
I do. Thanks. Beautiful talk. I really loved it. I'm interested in, so your splicing reporter is working so well that you might imagine that at some point you could learn the rules to be able to predict kind of which variants, so help with variant interpretation. Is that realistic, or is the search space, the sequence space in introns just so huge that that's a pipe dream? We don't have introns in yeast, so we don't have many of them.
So the introns make up 35% of the human genome. It's an incredibly vast space. So there's limitations with what we're doing, based on the limits of what you can actually synthesize cost effectively. In terms of oligo libraries, you're looking at 170 nucleotides. You can get longer oligos, but not at scale. And so 170 nucleotides, that gets us, after we factor in like the information you need for cloning and this multiplex barcoding, you get about 100 nucleotides of intronic information. That's not a ton. For really potent splice disrupting variants, it clearly works. You're going to miss a lot of stuff. And the reason that we started this, there are predictive tools, like computational tools out there for predicting splicing. I should mention that of all the really, really potent splice disruptors that we've identified empirically with experiments, none of those had impressive scores with those predictive tools. So what's clear is they can be very helpful, but they are not the solution to everything. You get you have an incredible amount of false negatives when you go down that route. And I just realized that rather than sitting around waiting or trying to build the perfect model, if we could just trudge through all of them with experiments, we'll just see it rather than trying to engineer something to tell me, let's just look.
Great. Thank you.
Mm-hmm.
All right. Thanks very much, Scott. We really appreciate the awesome seminar. And thanks for everyone for attending. We have another Science Matters coming up on November 8th with Danny Miller from the University of Washington. And thanks again to Scott.
Great. Thank you all very much for the invite. It was great to talk with you.